Okay, so today we're going to start with October-November 2015, paper 21, which is of 1 hour and 15 minutes. So the first question, figure 1.1 is an electron micrograph of mitochondrion. State the function of mitochondria. Mitochondria is the powerhouse of the cell. It generates ATP molecules for energy or aerobic respiration. Name the structure A. Now the structure A is the cristae of the mitochondria. Cristae. Okay, C. Calculate the magnification of the image in figure 1.1. Now we know that this actual length is equal to 0.7. The observational length would be in centimeter around 3.1, 3.2, 3.3 centimeter. Now we know that 100 centimeter is equal to 10 to the power 6 micrometers. Therefore 3.3 centimeter would be x micrometer. X would be equal to 3.3 into 10 to the power 6 divided by 100, which will give you 33,000, sorry. Now the formula of magnification is magnification is equals to observational length over actual. Now these two have to be in the same units, so we converted this into micrometers. Now actual is 0.7 micrometers and observational was 33,000 micrometers. If we divide this, we're gonna get the magnification to 33,000 divided by 0.7 micrometers will give you 47,142.85 magnification. Okay, so question number 1, part D, explain why the light microscope could not be used to produce the image in figure 1.1. So the light microscope, as we know, has a lower resolution, of about 200 nanometers as compared to the electron microscope, which has a resolution of about 0.5 nanometers. So the difference in resolution, which is the ability to distinguish between two distinct objects, is the reason that the image, the clear image, which is distinct, can only be seen with an electron microscope. Okay, part E, scientists think that mitochondria were once prokaryotes. The evidence for this is that mitochondria have features in common with prokaryotes. State 2 features that mitochondria have in common with prokaryotes. So one of the features is 70S ribosomes, which is a smaller subunit of ribosomes. They're smaller in size. And the second would be circular naked DNA. Those are the things in common. Question number 2 is a diagram of a protein channel for ions in cell surface membrane. Figure 2.1 shows the channel when open, and figure 2.1 shows the channel when the same channel is closed. Name the process by which ions pass through the membrane using channel proteins. So this is known as facilitated diffusion, which means diffusion which is helped, facilitated in a way. Part 2, explain why a channel protein is needed for ions to pass through cell membrane. Ions are firstly polar in nature. so they cannot pass through the non-polar bilayer. Secondly, the ions are too large to simply diffuse through the phospholipid bilayer. Thirdly, the ions would be hydrophilic, which means the same thing as polar in nature, and will be repelled by the hydrophobic layer. Now, since this is of 2 marks, you can earn any 2 valid points. Question number 2, part B. The channel protein figure 2.1 is made from identical polypeptide chains. Name the level of protein structure which is present when 5 polypeptide chains form the protein. So this would be the quaternary structure. The quaternary structure is when 2 or more than 2 protein polypeptide chains join together. Quaternary structure, or the 4-degree. The second part is the part labeled C. So let's see this. The part labeled C is this part. So as we can see, this is an alpha helix. This is an alpha helix chain, or made by the secondary structure. Okay. Channel proteins are example of transmembrane proteins. The polypeptides are held together and also interact with phospholipids in the membrane. Suggest how polypeptides are held together and suggest how they interact with the phospholipids. So in this question, they're asking us about the structure of the polypeptide chains. The structure of polypeptides is very basic. It's held together by 4 basic bonds, which are in pointers hydrogen bonds, peptide bonds, disulfide bonds, hydrophobic bonds between R groups, and lastly ionic bonds between charged polar groups. Disulfide bonds are held together through the sulfur molecules present in the chain. Peptide bond is a simple bond joining 2 of the amino acids together, which make up the polypeptide chain. Hydrogen bonds are between the nitrogen present in the polypeptide chain and the hydrogen present in the polypeptide chain. Sorry, this would be the nitrogen and the oxygen in the polypeptide chain in the basic structure. So these are all the 4 bonds that hold the polypeptide, and this is how they interact with the phospholipids through this structure. Figure 3.1 is a light micrograph of cells and root tip of a garlic plant. It is a diploid number 2n of 60. Diploid is the number of full chromosomes. So part A, name the stage of mitosis shown in cell D. So the stage in mitosis is this one, which is anaphase. where the chromosomes are where the chromatids and the sister chromatids are separated and move to the opposite poles. So explain why mitosis occurs in plants such as A. sabatum. So this is general question. Mitosis occurs so that growth is possible. Mitosis occurs so that reproduction is possible. Mitosis occurs to replace dead or worn out cells, etc. Now, since this is of 2 marks, you cannot write any valid 2 points. State the haploid number of A. sabatum. So the diploid number is 16, so the haploid number would be 8. Question number 3, part 2. Explain why a plant such as A. sabatum produces haploid cells. So it's not a part of the 2022 AS level syllabus, so no, we're not going to do this. Moving on to part D, DNA replication must occur before cell division. Describe the process of DNA replication. So this is of 5 marks, which requires very good detail. So firstly, DNA helicase acts on the double helical structure. This unwinds or unzips the double helix structure then comes DNA polymerase. The job of DNA polymerase is to add free nucleotides to the strands, to the unzipped strands of the DNA molecule. Now the DNA molecule has specific bases, for example adenine and thymine. Therefore, the nucleotide bases added are specific to the bases or complementary to the bases on the strand. Now the DNA polymerase can only copy in the 5' to 3' direction. Therefore, a leading strand is formed when the DNA strand runs in the 3' to 5' direction, and the lagging strand when the DNA strand runs in the 5' to 3' direction, which causes Okazaki fragments to form. Now comes the job of DNA ligase. The job of DNA ligase is to bind the Okazaki fragments together and to join the nucleotide bases together through phosphodiester bonds. This is how semi-conservative replication takes place. Semi-conservative DNA replication takes place. Question number 4. Figure 4.1 is a light micrograph of a section through a leaf of marram grass adapted to reduce transpiration. State what is meant by the term transpiration. Transpiration is a process through which water from the spongy mesophyll cells in the leaf evaporates into the substomatal air spaces and then diffuses out of the stomata into the air out of the leaf itself down a water potential gradient. Okay, so one adaptation of a arenaria is a curved leaf. Give one other adaptation and explain how this reduces transpiration. So one other adaptation of this leaf would be visible, that is the sunken stomata inside. So this would be sunken stomata. Explanation. The explanation for this would be that the stomata are inside the leaf. Therefore, there is lesser surface area for water to diffuse out, reducing transpiration. And since it's inside, there would be no wind affecting the rate of transpiration. Same question part B is saying that to investigate rates of transpiration of plants P and Q over 18-hour period. The results are shown in figure 4.2. So use figure 4.2 to calculate the difference between rates of transpiration and water absorption for species P at time 1400. So the rate of transpiration would be approximately 60 and the rate of water uptake of species P would be around 42. Now it wants the difference, so 60 minus 42, which would give us 18. Don't forget to write the units per unit mass over GH. 18 mass over GH. So the next part is describe and explain the patterns of transpiration and water absorption of species P. So in this we have to describe this graph, especially species P and the water uptake by species P. So the node pointers for this would be that at 0600 hours both of them have the same water absorption and transpiration rate, water uptake and transpiration of about 2.7 mass over GH. The transpiration rate slowly increases with increase in time, probably due to more sunlight acting on the leaves. And then becomes highest at 1400 hours of about 60 m/gh, after which it steadily decreases till 1730 hours of about 50 mgh, and then drops to 0 at 2400 hours. The rate of water uptake increases, and then it becomes highest at 1730 hours. Sorry, this is 18 hours and the graph 1800 hours in the graph, highest at 1713 hours, and has a rate of 51.7 approximately, and then steadily decreases to 3 mgh at 2400 hours. Now along this it says explain to. So an explanation for this would be that due to increase in sunlight, there would be an increase in transpiration rate, because there would be an increase in photosynthesis, there would be an increase in water usage, there would be an increase in the uptake. Therefore, there would be an increase in the stomata opening up for transpiration. So this would be the explanation for this question. Okay, so part 3 is suggest why the pattern of transpiration for species Q is not the same for species P. So if we look at this graph, we would see that species Q has a lower water uptake and transpiration rate as compared to species P, which is this. So a reason for this would be that species P is a xerophyte. Now xerophytes are those plants that are adapted to reduce transpiration or minimize transpiration during the day or due to a windy season, etc., etc. Now species Q would not be a xerophyte, and therefore it is not adapted to the circumstances. To the circumstances, and hence the transpiration rates are low. I'm sorry I mixed this up. Species Q is a xerophyte and species P is not a xerophyte, because species Q has a lesser rate of transpiration and species P has a greater rate of transpiration. So species Q is a xerophyte and species P would not be a xerophyte. So because of the adaptations, the transpiration rate of species Q is low and the transpiration of species P is high. So that would be this answer. Okay, so next question, part 5a, name the causative organism pathogen of measles and describe how measles pathogen is transmitted. It's not part of 2022 syllabus anymore, so we're going to skip this. Part C, the measles pathogen contains a single-stranded RNA molecule and no DNA. The cells that the measles pathogen infects contains double-stranded DNA molecules. State 2 other ways in which RNA in the measles pathogen differs from the DNA in the infected cells. So we know that the RNA is not a double helical structure, and it is a single strand. And secondly, we know that the bases in RNA are guanine, uracil, cytosine, and adenine, whereas the bases in DNA are guanine, thymine, cytosine, and adenine. So the difference is the uracil base RNA has a uracil base, whereas DNA has a thymine base. So that's the differences between RNA and DNA. Okay, so same question, part D would be explain why the measles pathogen cannot use an enzyme from the cell to carry out RNA replication. First of all, RNA replication is not done in the cell. It's made, however, it's not replicated. Therefore, there are no enzymes that can replicate RNA in the cell. RNA polymerase only makes new RNA strands. And secondly, even if there were strands, the enzymes in the cell would not meet the specificity of the measles pathogen to carry out the process, meaning that it would not have a complementary shape to the RNA replication process in the pathogen. Okay, so part E would be the outer part of the measles pathogen contains an antigen called the H protein. This antigen appears on the surface of cells infected with the measles pathogen. Describe the role by T lymphocytes in the primary immune response to an infection by the measles pathogen. Now, firstly, you should know that T lymphocytes are of two types, which are the helper cells and the killer cells. Let's talk about the helper cells. Helper cells, when they detect an antigen complementary to it, it divides by clonal selection and expansion by mitosis. Now, since this is a primary immune response, some of them make memory cells that persist in the body for a secondary immune response, and some of them become other cells which in turn secretes cytokines, which is a chemical that stimulates specific B lymphocyte cells to make plasma cells and memory cells complementary to the antigen, which in turn the plasma cells of the complementary B lymphocytes secretes antibodies which act against the pathogen, which is the H protein. Now the infected cell is displaying the pathogen on its surface in the question. This antigen appears on the surface of the cells infected with the measles. So the cells infected are displaying the pathogen on its surface, which is known as ACP, antigen presentation. So antigen cell presentation. So the T killer cells detect this infected cell and they make holes in it, in the cell wall, in the cell membrane, so that the infected cell bursts, so that the pathogen is removed along with the cell lysis. Lysis means to burst, bursting. The T killer cells makes holes in the cell membrane by producing toxins, and some of them, since it's a primary response, as the question says, they will make memory cells as well for a secondary immune response. Okay, so question number 6 would be figure 6.1 is a food web. This is not part of the 2022 syllabus, therefore we will not do this question.